We're now moving on to our latest partner on the platform. That being said, we're very happy to hear our corporate, our champion Behzad Mahdavi, the Vice President Strategic Innovation and Alliance for Pharma and Biotech. He will give us some insight in what the vision for the batch is and what they want to accomplish. So Behzad, welcome to you.
Thank you very much, Fabian. I'm very happy actually to be part of this program, and a special thanks actually to Plug and Play and to all the startup for their participation. So I would like to present the Lonza Pharma Biotech, a worldwide custom development and manufacturing leader in pharma, cell and gene therapy space. Our mission is to support our customers composed of large pharma and mid and small biotech to develop and manufacture the drug efficiently using advanced platform for their patients. One important element also on this page is the age of Lonza organization, 123 years. And the secret of remaining in this business in a successful way during so long is a constant evolution based on innovation. Basically, this innovation spirit is reflected in our motto, which is delivering the medicine of tomorrow today, and really mean it and do it. Today, we are supporting more than 1,000 therapies based on different platforms, from small molecules up to the cell and gene therapy, passing by biologics. We love working with external companies, and especially with startups that I love personally, and our partnership with Plug and Play is to find the next innovative solution together with our external startups. One of the high-profile example to illustrate this external partnership with a startup is when Lonza and industry actually in general had a challenge for the complex manufacturing of CAR T-cells therapies, where the T-cells, the soldier of our body, are taken out of the patient and genetically manipulated to recognize and kill the cancer cells and put back into the same patient. Thanks to our collaboration with a Canadian startup called Octane, we have been able to develop an intelligent and automated platform and bring the solution to the market. And the first patient has been injected in September, actually, with a product coming from this system. And today we have a whole new business unit created based on this initiative. The next challenge that we are looking actually in this coming batch is one around the exosomes, which are acting as FedEx of our body for the cell-to-cell communication. And they can be used both for therapeutics and drug delivery, as well as companion diagnostic. And our targets are the startups with new technologies to manufacture efficiency and load in an efficient way this type of exosomes. The second subject is around more cell line engineering for complex protein expression technology that support manufacturing of B or multi basically specific antibodies in mammalian cell and without any having a folding or misfolding aspect to that. And we have, of course, the two excellent startups that Fabian is going to present to us in the few minutes, actually. Thank you.
All right, Behzad, thank you so much for the insights, and also for the insights on the vision for the upcoming batch. It's great to have you on board, and it's also very exciting for us to finally kick things off during the program with the partnership and see where you go with the next two startups which are upcoming. So first of all, we will see MD Immune. They are based out of Seoul, and they've developed a drug delivery platform called BioDrone. Using their vesicle, they yield a 10 to 30 times production rate compared to normal exosomes. How they do it, Seong will tell you more about it and guide you through the company.
All right, thanks, Fabian. Hope you can see me okay, and we'll start the PowerPoint. Okay, here we go. So hello, everyone. My name is Seong. I'm a CSO at MDimmune. I'm very honored to be part of this exciting event. MDimmune, sorry for the delay. Okay, MDimmune is a biotech company headquartered in Seoul. We've been dedicated to the development of the innovative therapeutic platforms called BioDrone. So this video clip actually illustrates the drone delivery project at Amazon, which is, I thought was very fascinating to watch. So coming out of this model drone, large blimps can be sent out with hundreds and thousands of packages to deliver them to the exact location. And I think the potential for the drone delivery to revolutionize the courier services is just immense. And I believe our platform technology resembles this drone delivery in many ways. BioDrone platform relies on the proprietary extrusion method to obtain the tiny vesicles from cells. These cell-derived vesicles, or CDVs, are then just used as novel, nano-sized drug delivery vesicles. After loaded with many types of drugs, these vesicles can find the targets, tissues, or cells inside our body using the interaction between particular surface moieties and their binding partners and targets. The first step is extrusion. Cells in suspension are passed through membrane filters with narrow pore sizes. As the pore size progressively decreases, finally to under a micron, the cells revascularize into tiny nanovesicles, which are very similar to exosomes in size, shape, and sharing many biochemical properties. If we extract CDVs from naturally available cells, such as mesenchymal stem cells, CDVs are produced with properties similar to their parent cells, or even exosomes secreted by MSCs. But what is more exciting is the surface modification or other engineering is possible, thereby conferring many interesting characteristics on the CDVs. These include a precise homing capability or characteristics enhancing therapeutic potentials. Currently, this extrusion technology is patented in 5 different regions. One exciting feature of CDVs is that they can be loaded with multiple drugs. This interesting quality has been demonstrated by many EV researchers using both chemical and biomolecular payloads. Using one of the available methods, we have observed highly efficient RNA loading into the CDVs and subsequent knockdown of target gene expression. The other feature that substantiates the potential of CDVs is that they can be steered to find the target. While we've been focusing at the initial stage has been the interaction between cell adhesion molecules, such as integrin and its binding partners. In addition to the natural cell sources, we are also exploring the possibility of enhancing such targeting effect by overexpressing surface markers on the source cells or combining other emerging technologies, which is well suited to CDVs. Next, let me briefly summarize the unique advantages of the BioDrone platform technology over existing drug delivery systems. Since CDVs are made from natural human cells, they are safe and can penetrate cell membranes and tissues fairly easily. In other words, the body is less likely to reject them in the same manner as it does other conventional drugs or other synthetic vehicles. In addition, we have observed that CDVs can effectively penetrate the blood-brain barrier. Therefore, using this property, highly potent but toxic CNS drugs could more easily be used in treating the brain diseases. Another remarkable feature of CDV is their versatility and expandability, which exosomes do not currently exhibit. BioDrone platform technology can be applied to virtually any cells. Moreover, engineering source cells that express surface proteins on the plasma membrane is so much easier than attempting to do the same on the membranes of exosomes. As shown in the left figure, we have observed that CDVs loaded with membrane proteins important for muscle differentiation, for example, could rescue muscle phenotypes in the cells deficient of such proteins. Finally, compared to the exosomes, we can produce far more nanovesicles, nanoparticles in a much quicker process. This video clip shows how our syringe type extruder we developed works. it can process up to 200 milliliter of cell suspension in one click, which results in yield that is significantly higher than conventional exosomes purification methods. Besides the aforementioned versatility and flexibility, we believe that BioDrone platform technology will be expanded in many ways and help cure many debilitating diseases that are resistant to conventional therapies. So I guess I should wrap up for the sake of time with many thanks to the organizers at the Plug and Play, and of course our sponsor Lonza, and the audience, and most importantly, all the members of the Exosome, MDimmune that I'm so proud of. Thank you.
Thank you, Sion. Really on point. So we already had the first questions coming in. Well, the first one is, how did you test your word error rate against other solutions?
Pardon? Which rate?
How did you test your word error rate against other solutions?
I'm sorry, I didn't understand the question quite well.
Okay, then maybe quickly jumping to the next one. In that specific case, what is the process for correcting any errors in transcription?
So it depends on the cargoes that we would like to load. So it can be genes, and I mean, it can be the small RNAs or bigger molecules, or it can be chemical drugs. And so, and I mean, that's how it works on the CDVs that we are trying to develop.
Okay, fantastic. Thank you very much. Well, is the targeting as specific as for viral vectors?
So that's an excellent question, and then we are really diligently testing and comparing with other drug delivery systems. But since we are based on the natural the interactions that the cells and the body has, so by expressing them, I think we believe that we can achieve the similar or more effective targeting capability as shown in the virus or other vehicles.
That's fantastic. Well, Sion, one question from our side. What do you expect from the Startup Creasphere program? What do you want to accomplish with us and also with Lonza together?
All right, so I believe that by partnering the prestigious company like Lonza, we believe that we can escalate our technology to the next level that is more close to the commercialization level, and also try to understand what the global pharma or mature stage companies would like to see from us.
Fantastic. We're very looking much forward to working with you together and to see where the batch takes us. So thank you very much, Sion.
My pleasure.
All right, then last but not least, I'm happy to introduce you to Valitacell. Valitacell has developed a technology that enables biologic drug manufacturers to bring their products to market much quicker. How they do it and what's the product offer, Stephanie will guide you through that.
Thank you, Fabian. Good afternoon, everyone. Great to be here. I've really enjoyed the presentation so far. I'd like to start by thanking Plug and Play and Lonza for the opportunity to present to you all, but also to engage with the program. My name is Stephanie. I'm the head of Science Cell Therapy at Valitacell, and I'm here just to introduce you briefly to the company. So Valitacell is an Irish biotech SME that's headquartered out of Dublin at the National Institute of Bioprocess Research and Training, with a cell therapy R&D program at the Regenerative Medicine Institute in Galway and manufacturing capabilities in Germany. Now, the primary goal of our company is to bring technologies to biologics and cell therapy manufacturers that allows them to bring their products to market faster, cheaper, and with greater regulatory confidence. So quite a bit of synergy with Lonza's mission. Essentially, biologics or protein drugs such as monoclonal antibodies and cell therapies offer a really exciting new modality for the treatment of many different kinds of diseases, from certain types of cancer to degenerative diseases like osteoarthritis. However, there are significant engineering and manufacturing challenges that need to be overcome to bring these therapeutics to the patient and through the clinic. At Valitacell, we really believe that better analytical technologies are required to really drive the evolution of this manufacturing process. Essentially, better product characterization or enhanced product characterization will drive the process control that is really needed or key to success. Within the biomanufacturing process, we target 3 core components with our technologies: the product or the protein product itself, the cell that is either used to produce the product, or in the case of cell therapy, that is the product, and the media in which the cells are grown and manufactured. We have 4 technology platforms that we have developed to target various elements of these 3 components. I'll start by describing ChemStress, which is a functional characterization tool for both CHO and mesenchymal stromal cells. It's a 96-well plate-based assay that uses small molecule stressors to profile cell response in specific media environments, and can be used to functionally characterize your cells throughout your process development. Quantum is a fluorescence polarization-based platform that is used for protein or target quantification or detection in solution. Enhance is a media design platform that we use to customize or optimize media formulations for the biomanufacturing process, specific to your process. And Cell AI is an image-based platform that really harnesses the power of AI to characterize cells based on specific features of the cell morphology. So the overarching goal of these 4 technology platforms is to put useful information into the hands of biomanufacturers as early as possible in their process. So Valitacell was founded in 2015, and since then we have successfully commercialized 2 of these platforms, including ChemStress and Quantum, for the biologics space, which with blue chip customers across European and US markets. And we are currently developing both ChemStress and Quantum for the cell therapy space, touching on maybe EV or exosome quantification with Quantum that's been discussed previously. We are then also developing Enhance and Cell AI for both the biologics and cell therapy space, with the aim of commercializing these in the coming years. I'm going to leave you with just a snapshot of our senior team, because we really feel that key to our success thus far and to the delivery of the technologies that we have in development is the team that we've assembled at Valitacell. We've got over 45 years of collective experience in the life sciences industry, a significant track record in tech development and commercialization through our CEO, Jerry Clifford, as well as domain expertise in areas like AI through our CTO and head of data, Ben and Paul. Myself and my colleague Hannah bring cell therapy and biologics manufacturing expertise to the group. And we also have core competencies in image-based assessment of cells through my colleague Eligio. And Kevin, who's actually here with me today, is our Chief Marketing Officer, who has 20 years of experience in growing brands across multiple sectors and is leading our digital marketing campaign in the space. So this is just a snapshot of who we are and what we do, and I'm happy to answer any questions.
Thank you, Stephanie, for that great presentations and all the insights. Well, we already have the first question, and it is about the accuracy of your quantum platform. So with which accuracy can you actually say you've identified the target?
So our current products in the quantum platform target monoclonal antibodies, and we have benchmarked these against established technologies in the space like the octet and HPLC. And it is a relative quantification tool, so requires a standard curve, but works very well with high accuracy. And I think the advantages of the technology over larger instruments like I've just described are really that it is quite high throughput, doesn't require specific hardware or specialized hardware, excuse me, and can also be, we are in the process of migrating it towards an at-line application. So it's very, very dynamic and flexible, but also simple and easy to run.
Sounds fantastic. Well, on the other side, also looking at your entire product portfolio, where do you see the highest traction in the future, or what do you believe will be the most successful product?
Personally, I think there's space for all products at different points of the development and manufacturing process, but the Cell AI platform has a lot of potential in the cell therapy space, which is really lacking, I think, tools that allow for quick turnaround of information and manipulation of your process. At the moment, most analytical platforms take a long time to characterize the cells and don't allow for manipulation of your process. So I think AI-driven image-based analysis really has a lot of potential in this space.
Maybe also directly touching base on the cell AI. So how much data have you already accumulated in order to develop this product?
So part of our goal really is to acquire data through all of our projects to feed into the Cell AI program. And we're very lucky in that we have a number of collaborators and test beds across Europe in both the biologics and cell therapy space that have allowed us to do that. So we've acquired quite a bit of data so far, and we've generated a number of proof of concepts, both in mesenchymal stem cell morphology assessment and CHO cell viability assessment as well.
Okay, fantastic. Then one last question. What do you expect? How much time can be saved using your solution in setting up, well, a production process?
Well, if we take the example of AI, and you suggest that you don't need to extract cells from the process and stain them to get valuable information, you reduce assays that normally, especially in the mesenchymal stem cell manufacturing setting, for example, could take days to minutes and hours. So it really has the potential to really optimize the process, because you can capture information much faster than before.
Very impressive, and might have some great influence in the industry. Well, thank you so much, Stephanie and Kevin. It was great to have you, and thank you for all the insights and answering all those questions. Well, and then also thank you from my side. It was a pleasure to host all the startups, to welcome all the startups. We're really excited to kick our batch 5 off. And we're looking forward to see the developments in, well, in 12 weeks from now. That was it from my side. I'm now handing over again to my colleague Federica for the closing remarks. Thank you very much.
Okay, perfect. Thank you very much, Fabian. And before the closing remarks, just one brief announcement. Today there was a press release from Roche Diagnostic and our batch 2 startup BabyScript that they have come into a commercial agreement and are starting their collaboration continuing after our batch. So congratulations. And we have our next event upcoming, the batch 5 Expo, where you can see what the startups of this batch have achieved during the batch, starting as of today. And hopefully we have a lot of more press releases like this afterwards. So I would like to thank everyone for participating, for asking questions, for pitching, sharing what you do in your company. And we really have so many different important angles of the healthcare industry present with the same goal to transform healthcare together. Thank you to our dear partners for supporting us and for supporting the startups. Good luck to the startups and your pilot projects. Thank you for participating in our program. We're very proud and happy to have you on board. And most importantly, thank you to the team here on the ground from Plug and Play for preparing and for executing this event. We are all not born AV and tech experts, and I think we can be very proud of us, of what we have achieved. We had over 200 participants today, and I would like to thank everyone for joining us, and big thank you to the team here on the ground and everyone for joining us, and looking forward to a successful batch 5 together. Thank you very much.